Good afternoon, everyone. Today, we are going to discuss synopsis and questions of a one more chapter, which is known as biomolecules. Biomolecule chapter is discussed under zoology concept, where it is going to deal with the availability of different variety of biomacromolecules in the nature. So that is referred to as the biomolecule. So here this biomolecule chapter mainly concentrates on the availability of different variety of macromolecules in the nature. At the same time, the monomers that are involved in the formation of the macromolecule. So that is the point lying behind in this chapter. This is divided into two parts. Biomolecules chapter is divided into two parts. One is the monomers, second one known as polymer. Under monomer, we need to know about amino acid, sugars, fatty acid, glycerol, nucleotide, nucleoside. These all belongs to the biomolecules. That is, the monomers are present here. Okay. Monomers has been given here. So when you come to the polymers includes proteins, carbohydrates, lipids, nucleic acid. This is how we are going to make classification under biomolecules. The third classification, enzymes. Okay, third one is enzymes. Under enzymes, we are going to know about classification, activation energy, factors that affect the role of enzymes. Okay. Enzyme activity. This is the general outline of the chapter biomolecules. First, let me know about what is biomolecule, what are the examples that we can quote for biomolecule, and which are the things that are included under the biomolecules. Okay. The molecules which are found inside the body of a living organisms are said to be as the biomolecule. Okay, the components that are available inside the body of an organism is said to be as biomolecules. So just quote with that. Biomolecules. Molecules found in the cytoplasm of cells are said to be as biomolecule, which includes hydrogen, carbon, nitrogen, oxygen, sulfur, phosphorus, etc. Biomolecules are again classified into two types, bio-micromolecules and bio-macromolecules. Okay, it is classified into micromolecules, macromolecules. The monomers are said to be as micromolecules. Okay, the monomers are said to be as micromolecules. Okay, polymers are said to be as the macromolecule. Amino acid is a micromolecule. Amino acid joins together to form polymer that is called as the macromolecule. So how we are going to analyze? Okay, how we are going to do? It is by chemical analysis. Chemical analysis of the biomolecules. How we are going to analyze the molecule? First one is take the tissue, okay, add acetic acid, trichloroacetic acid, grind, filter, we get two medium, acid soluble pool, acid insoluble pool. Acid soluble pool. The acid soluble pool contains the micromolecules. They contain the micromolecules. Okay, acid soluble pool contain micromolecule, whereas acid insoluble pool contain macromolecules. The examples for them are macromolecules are larger in size, they have a high molecular weight, they are formed by condensation of the micromolecules. So here they are greater than 1000 Daltons. They are less than 800 Daltons. Okay, they are greater than 1000 Daltons. When compared to the lipids, lipids are even less than 800 Daltons. They are still more less than 800 Daltons. Okay, so based on this, we are having the primary metabolites and secondary metabolites. Primary metabolites as well as secondary metabolites. The products that are formed by the physiological processes of the plant are said to be as primary metabolites. ಅರ್ಥ ಆಗ್ತಾ ಹೇಳ್ತಿರೋದು the organic compounds that are primary metabolites are those which are obtained by the physiological processes, which are obtained through physiological processes. They are said to be as primary metabolites. Carbohydrates, proteins, fats are said to be as amino acids. Nucleic acids are said to be as primary metabolite. When we come to secondary metabolite are those which are derived from primary metabolite, which are derived from primary metabolite. So it includes ಏನೇನಿರುತ್ತೆ ಅದರಲ್ಲಿ example gums, oils, resins, oil, resins, rubber, drugs, etc. Okay, na? So for example, some of the secondary metabolites are, when you come to the pigments, we have anthocyanin, the red pigment, okay, carotenoids, orange pigment, alkaloids, morphine and codeine, terpenoids, monoterpenes, diterpenes, essential oil, lemongrass oil, toxin, arbin and resin, lectins, concavalin. Concavalin is a lectin. This is important. Mark this. Based on this, they're going to pose the question. Concavalin A. This is a lectin. Drugs, vinblastin, curcumin extracted from the turmeric, vinblastin, vincristin are anti-cancerous drugs which are going to treat the cancers. Okay, so like this we are having primary metabolites and secondary metabolites. Under chemical analysis, we can extract the primary and secondary metabolite. There is one more method of analysis, finding out the dry weight, where the biomolecule is burnt and it is dried and its weight is analyzed. That is called as ash method, where we are going to weigh the ash and we are going to find out the molecular weight of the biomacromolecules. This is a different methods that we are going to use the biomacromolecule analysis. Okay, next we shall know about micromolecules. First micromolecule is amino acid. Amino acids are the monomer of the proteins. Amino acids joins together to form a polymer called proteins. They joins together to form a polymer called as proteins. Amino acid contains amine group plus carboxyl group. It is made up of amine group and carboxyl group. We call it as alpha carbon, where a carbon is surrounded by 4 different variety. One carbon is surrounded by 4 different functional group. For that we call it as alpha carbon. So if you want to see the structure, this is the carbon. The carbon is surrounded by amine group NH3. It is having carboxylic group COOH. Here we are having hydrogen and here we are having the functional group R. So one carbon associated to 4 different groups is said to be as alpha carbon atom. Alpha carbon atom. So if you observe this, these amino acids, they are of different varieties, like we have acidic amino acid, basic amino acid, neutral amino acid. Acidic, basic, neutral, different type of amino acids. If it is having more than one COOH group, then it is called acidic amino acid. If it is having more than amine group is said to be as basic. If it is having one amine plus one carboxyl group, then we call it as neutral amino acids. There are different variety of amino acids, essential and non-essential amino acid. The amino acids which are required for our body, which are not required in our body, are said to be as essential amino non-essential amino acid. The amino acids which are required for our body are called essential amino acid. 20 different variety of amino acids are there. Linolenic acid, linoleic acid, linolenic acid are the olic acid, linoleic acid, linolenic acid are the three different types of amino acids that are present, that are required for our body. Olic, linoleic, linolenic. All these three are said to be as the essential type of amino acids. Are three different type of essential amino acids. These amino acids group together to form proteins. Proteins are made up of amino acids. N number of amino acids group together by peptide bond. One amino acid linked to another amino acid with the loss of water, formation of NHCO bond. For loss of water with the formation of NHCO bond is said to be as peptide bond. The proteins are arranged in different patterns. There are four types of proteins we are going to observe. They are primary protein, which are linear in nature. Secondary protein, which are having alpha helix, beta plated structure. This is called alpha helix and this is called beta plated structure. Tertiary. Tertiary structure includes three-dimensional, highly coiled structure. Okay, when it comes to the last one, quaternary. Quaternary is formed by combination of two conjugate acids connected with the help of disulfide bond. That's called as quaternary type of amino acids. So primary amino acids is observed in all type of amino acids. Secondary amino acids is formed by, it is observed in keratin of hair as well as pleated structure in silk. This is found in keratin. This is found in silk. Whereas tertiary structure is most established structure. This is observed in many varieties of enzymes. In case of quaternary structure, it is observed in case of hemoglobin. Hemoglobin possesses quaternary structure. Here it has two subunits, 2 alpha, 2 beta, joined together by the disulfide bond that forms quaternary structure. Okay. Next is, which are some of the important proteins that's involved in functioning is GLUTA4, glucose transporter, Glucose transport 4 enables the transportation of the glucose. Antibody receptor fight against infectious agent. Collagen. Collagen is the most abundant structural component, ground substance. Trypsin is the enzyme. Trypsin is the enzyme involved in protein metabolism. Insulin is the hormone. So let's move on to carbohydrates. Before entering into the carbohydrates, let's know about monosaccharide, disaccharide, then comes to polysaccharide, carbohydrates. Carbohydrates are made up of carbon, hydrogen, and oxygen. They are the oxides. They are the oxides of the carbons. They are having the formula CnH2nOn, wherein refers to a number of carbon atoms. Based on the number of carbon atoms we have classified. The simplest one is called monosaccharides. Saccharides means sugars. Monosaccharide, disaccharides, polysaccharides. Three different varieties are observed. Monosaccharide is made up of 3 carbon, 4 carbon, 5 carbon, 6 carbon, 7 carbon atoms. Okay, so 3 carbon is xylulose, triosus. Triosus is the 3 carbon, tetrosus is 4 carbon, pentose is 5 carbon, hexose is 6 carbon, heptose is the 7 carbon. Okay, so triosus. So triosus, tetrosus, heptosus, hexosus, and heptosis are the different variety of carbons that we are going to observe. So ಇಲ್ಲಿ ನಮಗೆ 4 carbon ಗೆ ಬಂದ್ರೆ ribulose, xylulose are observed. Pentose ಗೆ ಬಂದ್ರೆ ribose, deoxyribose. Hexose ಗೆ ಬಂದ್ರೆ glucose, fructose, galactose. Like this we have the most. The fructose is the most common abundant. Fructose is the most abundant form of sugar. Okay, so next, the monosaccharides have some of the monosaccharide has a property to reduce Cu, copper 2 plus ions to copper plus ion. They are called as reducing sugars. If they want to have, if they are called as reducing sugars, they should have free aldehyde or ketone group. So sucrose is one of the best. Sucrose is one of the best reducing sugar. The sugars can be identified by a method called Benedict's reagent. So we need to take 2 ml of solution plus add 2 ml of solution plus 5 ml of Benedict's reagent plus heat it. It is going to show the change in color from blue to green, green to yellow, yellow to brick red, yellow to brick red. Blue color shows there is no accretion of sugar. Green color shows there is a slight secretion of sugar. Yellow color shows maximum amount of sugar is excreted through urine. Brick red shows maximum amount of glucose is excreted through the urine. Okay, so Benedict's reagent, Fehling solution, all are the different methods used to identify the carbohydrates. The bond present here is called glycosidic bond. The bond is called glycosidic bond. It may be alpha 1,4 glycosidic bonds. Alpha 1,4 glycosidic bonds. alpha 1 comma 6 glycosidic bonds. Sucrose is a non-reducing sugar. Lactose is 1 glucose and galactose. Fructose is also having glucose and sorry. So when you come to the monosaccharides are these are the things which we are going to say it as the monosaccharides. Next comes to the disaccharide. Sucrose, lactose, maltose. Sucrose is made up of 1 glucose and 1 fructose. It is identified in sugarcane as well as in sugar beet. 1 glucose, 1 fructose, lactose, glucose, galactose, maltose, glucose plus glucose is said to be as maltose. Maltose is the sweetest among naturally occurring sugars. So next third one is polysaccharides. Polysaccharides are made up of n number of they are made up of n number of monomers that is said to be as the polysaccharides. The polysaccharides has been divided into storage polysaccharide, structural polysaccharides. Okay, storage polysaccharide anta vandre which is involved in the storage of the food material. Under food storage polysaccharide, we have starch. Starch is a helical secondary structure. It has a capacity to hold the iodine molecule. Starch is abundant in rice, wheat, potato, okay, legumes, etc. It is formed during photosynthesis and it is used as an energy storage in case of the plants. So animals have glycogen. Animals have glycogen in animals. Okay, so storing carbohydrates takes place in the form of polysaccharide has two advantages. They can be broken down in any time and they can be reformed. Okay. In case of the polysaccharides, the right end is a reducing end, left ending is non-reducing end. Next comes to the structural polysaccharides. Under structural polysaccharide, we have chitin. Chitin is a polysaccharide found in the exoskeleton of insects, crabs, and prawn. It is made up or it is similar to the cellulose, but it is having N-acetyl glucosamine. Cellulose. Cellulose is made up of hexosan polysaccharide. It's the most unbranched polysaccharide. Unbranched homopolysaccharide. Fibers of cotton, linen, jute are used for textiles, ropes. Insulin is a polymer of fructose. Okay, so these are the different varieties that we are going to observe it under polysaccharide. Starch, homopolysaccharide. Okay, chitin, heteropolysaccharide. Cellulose, unbranched homopolysaccharide. Insulin, heteropolysaccharide, because it's made up of A as well as B chain. Okay, 20 and 31 amino acids are present totally together to form 51 amino acids are present here. Okay, so structural polysaccharides as well as the homopolysaccharide and heteropolysaccharides are observed in case of the carbohydrates. Okay, so these are the information regarding to the monomers as well as the polymers. So we completed with the structure of protein, amino acids, monomers and the carbohydrates. Next we shall move on to the lipids. Next is lipids. Lipids are made up of carbon, hydrogen and oxygen. They are made up of carbon, hydrogen and oxygen. The number of oxygen atom in the lipid molecules is always less than compared to the number of carbon atoms. Sometimes phosphorus, nitrogen, sulfur are also present. These lipids are insoluble in water, but they're soluble in solvents like chloroform as well as the benzene. Lipids are made up of fatty acids plus glycerol. Lipids are made up of fatty acids and the glycerol. Fatty acids are of two types. Fatty acids are of two types. Saturated fatty acid as well as unsaturated fatty acid, based on the presence of the double bond and the single bond. Saturated fatty acid. Saturated fatty acids are those which are not having the double bonds. They are having only the single bond, are said to be as saturated fatty acid. Unsaturated fatty acids are those which possesses the double bond between the carbon atoms. Okay, so palmitic acid, saturated fatty acids. Okay, phospholipid, lecithin is a saturated fatty acid. Cholesterol is the main precursor for all these varieties of fatty acids. Okay, when fatty acid combined with the glycerol, it results in the formation of glycerides. If one fatty acid combines with one hydroxyl group of glycerol, is called monoglyceride. If two fatty acid combines with two hydroxyl group, it forms diglycerides. If three fatty acid combines with three hydroxyl group of glycerol, then it is called as triglycerides. So monoglycerides, triglycerides and the diglycerides are formed by the combination of fatty acids and the glycerol. Fatty acids and glycerol. Okay, so phospholecithin. Lecithin is formed by the combination of fatty acid, glycerol and cholesterol in them. This lecithin is most abundant in our plasma membrane. Fats. Fats are the esters of fatty acid and glycerol. They are liquid at room temperature. They are said to be as oils, jingly oil. Okay. Phospholipid. Phospholipid is a type of lipid which is found in plasma membrane which has a phosphate head group and fatty acid tail groups. So what are the functions of these lipids? Lipids are rich in plants as well as in animals. They act as a storage component. They provide the high source of energy. Okay, next. Next is nucleic acid. Nucleic acids are responsible for providing genetic material of an organism. The nucleic acids are present inside the nucleus of an organism. These nucleic acids are DNA and RNA. DNA is the major type of nucleic acid present in all the organisms. They are present in all the organisms. That is the DNA. So what is this nucleic acid basically made up of? Nucleic acid is basically made up of nitrogen bases plus sugars plus phosphate group. Okay, so the nitrogen base, when it combines with the sugar, it will form nucleoside. Okay, when this side combines with phosphate group, then it will form tide. Nucleosides, nucleotides. Nucleotides joins together by phosphodiester bond to form nucleic acid. When you come to the nitrogen bases, we are having four different varieties of nitrogen bases. They are adenine, guanine, cytosine, thiamine, uracil. These three are considered to be as pyrimidines. These two are called as purines. Purines and pyrimidines are the two different types of nitrogen bases available in nature. Then it is combined with the sugar. If it is having HOH, it's called ribose sugar. If the region is having lack of oxygen, it's called as deoxyribose sugar. So the nucleic acids are the high energy compounds. When they break, they are going to yield the energy. Adenosine monophosphate, diphosphate, and triphosphate energy currency of the cells. Okay, these are the structures regarding to the nucleic acids as well as the nucleotides. Nucleic acid, when you come to the structure of a DNA double helix model, which runs always parallel to each other, they're having four nitrogen bases, which are connected by complementary based on the Charagov's complementarity rule. Okay, the one turn is of about 34 Armstrong. Distance between one nucleotide to another nucleotide is 3.4 Armstrong. Therefore, number of nucleotides is 10 nucleotides. It's the B helical structure DNA. Okay. Next is enzymes. Enzymes. Enzymes are the biological catalysts which are purely or basically made up of the proteins. They are basically made up of proteins are said to be as the enzymes. The term enzyme was discovered by Curie. Okay, it was discovered by Curie. Ribozyme is a type of enzyme that is present inside the ribosomes. Okay, na? All the enzymes are made up of proteins. Okay, they are called as simple enzyme. Simple enzyme made up of only proteins. When you come to the complex enzyme, it is made up of two parts. Protein part is called apoenzyme. Non-protein part is called prosthetic group. This prosthetic group, it may be inorganic or it may be organic. The prosthetic group is also called as three kind of cofactors are there. Protein part na apoenzyme ಅಂತೀವಿ. ನಾನ್ ಪ್ರೋಟೀನ್ ಪಾರ್ಟ್ ಅನ್ನ ಇದನ್ನ ನಾವೇನಂತ ಕರಿಬಹುದು ಅಂತಂದ್ರೆ cofactors ಅಂತ ಕರಿಬಹುದು. Cofactors are of three types. These three cofactors are ಮೊದಲನೇದು prosthetic group, ಎರಡನೆಯದು coenzyme, ಮೂರನೆಯದು metal ion. Prosthetic group ಅಂತ ಬಂದಾಗ that is completely made up of organic compounds, okay, which bound tightly to form protein part. Peroxidase involved in breaking down of hydrogen peroxide. Heme is the prosthetic group. Heme is the prosthetic group. Comes to the cofactors. They are the organic compound, but their association with the apoenzyme is only very transient. The essential components of many coenzymes are vitamins. Okay, here it includes the vitamins. NAD. Metal ion. Number of enzymes require metal ion for their activity. Zinc is essential to carry out carboxy peptidase. Next, activation energy. There is a one graph which explains about the activation energy. This is the level. This is activation energy without enzyme. Activation energy with enzyme. In the presence of enzyme, activation energy reduces. In the presence of enzyme, activation energy is reduced. In the absence of enzyme, activation energy is maximum. The substrate enters into the transient state at higher level. So this is referred to as the activation energy. The y-axis plots the substrate. X-axis shows the rate of reaction. Which are the factors that are going to affect the enzyme activity? Temperature. It requires optimum temperature in order to show the effective process of the activity. Then pH. Optimum pH is required in order to carry out the process. Substrate concentration. If there is a maximum amount of substrate concentration, rate of reaction will be also maximum. Okay, next is classification of enzyme. On the basis of the function performed by them, we can classify the enzymes into oxidoreductase. Oxidoreductase are the enzymes which are involved in the oxidoreductase are the enzymes which are involved in catalyzing either oxidation reaction or the reduction reaction of their substrate by removing or adding electrons. Okay, therefore that is called as oxidoreductase enzyme. Second one is called transferase. It is involved in transferring of a group of ion from one substrate to product or from one reactant to another reactant, results in the formation of the products. Hydrolases, they are responsible for breaking down large molecule into smaller molecule by addition of water, are called hydrolases. Next, lyases. Lyases are the structures that are going to cleave specific covalent bonds without hydrolysis. Next, isomerase. They are going to bring about conformational change in an atom or in a molecule. Glucose undergo conformational change to form the fructose. Glucose with the addition of phosphate will form glucose 6-phosphate in the form of phosphokinase. Next, ligases. They are going to join two molecules. Join two molecules. So these are the classification. Next is mechanism. The mechanism of enzyme action includes lock and key model. As we all know that lock and key model from your high schooling level, specific key can open a specific lock. Same way, specific enzyme can bind to the specific substrate and bring about conversion of substrate into products. Next is Koshland's induced fit theory. According to this, whatever the structure of the active site, after substrate binds to the active site undergo conformational change, takes the shape of the substrate and helps in conversion of substrate into the product. That's called as Koshland induced fit theory. These two are the different type of mechanisms. We are having inhibitors which are going to slow down the rate of reaction. One is competitive inhibitor and another one is called non-competitive inhibitor. Under competitive inhibitor, the inhibitor is having exactly the shape similar to that of substrate. They get confused for the substrate and it's going to stop the process. Non-competitive inhibitor, for example, cyanide. Cyanide kills an animal by inhibiting cytochrome oxidase, a mitochondrial enzyme, which is responsible for cellular respiration. Okay, cyanide kills the animal by interfering the cytochrome oxidase. Like this, we have different variety of inhibitors which are going to inhibit the process. So these are the synopsis regarding to the biomolecules. Next, we shall move on to the questions. First, all the macromolecules are results of the process of polymerization, a process in which repeating subunit termed monomer are bound into chains of different length. All the macromolecules are polymers except lipids. Which of the following are basic amino acid? Glycine, alanine, lysine, arginine, glutamic acid, and aspartic acid. Okay, histidine and proline. Basic amino acids possess two amine group, one carboxyl group, glutamic acid and aspartic acid. Glutamic acid and aspartic acid are considered to be as the basic amino acids. On losing carboxyl group as carbon dioxide, amino acids form biologically active glucose amines such as histamine, alcohol, nitrogen base. Okay, so here, what's the question? If carboxylic group is lost in the form of carbon dioxide, then amino acid become biologically active as nitrogen bases. Beta-plated structure of protein is present in silk fiber. Protein of these fibers are fibroin silk. Keratin of hair has, they said no, alpha helical structure of protein. Beta pleated structure in case of silk made up of fibroin. Most blood proteins in our body are basic. Casein of milk is a glycochromophosphometalle. It's a phosphoprotein. Lactose glucose and galactose. A monosaccharide is a simple polyhydroxyaldehyde or ketone molecule which cannot be further hydrolyzed into smaller unit. Number of carbon atom in monosaccharide may vary from 2 to 8. The sweetest of all naturally occurring sugars are fructose. Which of the following is not a reducing sugar? Sucrose. It doesn't contain a free aldehyde or a ketone group. Which of the following yield only glucose on hydrolysis? It's maltose. Storing carbohydrates in the form of polysaccharide has which of the following advantage? During their formation, many molecules water are removed from monosaccharide, condensing to be stored. When necessarily polysaccharide broken down by enzymes with the release of energy. Unlike small carbohydrates, polysaccharides are relatively small carbohydrates. Unlike small carbohydrates, polysaccharides are relatively easy to store. All of this, all of this is the answer. Most abundant organic compound in biosphere is cellulose. Yes. Largest amount of cellulose amongst natural material is present in wood, cotton fiber, roughage. Glycosidic linkage at a place of branching in starch and glycogen has alpha 1 gamma 4 glycosidic linkages. Monomer unit of glycan is N-acetyl glucosamine. Cellulose is a heptopolysaccharide, heteropolysaccharide branch, hexanopolysaccharide unbranched, pentasanopolysaccharide branch. It's a hexanopolysaccharide unbranched. Chitin is the second most abundant organic substance in the biosphere and is present in exoskeleton of insects and crustaceans. It is a protein, polysaccharide and basic unit of N-acetyl glucosamine. So option B. Which of the following is essential fatty acid with two double bonds in the chain? It is essential linoleic acid. Lecithin is a phospholipid with a choline attached to the phosphate head group. The backbone of nucleic acid strand is made up of sugar, phosphate, as well as sugar, base, and phosphate. Which of the following is not present in DNA? It is thymine. Spelling is wrong. Nucleotide. Nucleotides take part in information transfer system, energy transfer system, formation of NAD and FAD which acts as coenzyme oxidizing enzymes, all of this. A nucleotide is made up of sugar, phosphate, nitrogenous base, sugar, nitrogenous base, sugar, and phosphate. Nucleoside on hydrolysis will not yield sugar ಕೊಟ್ಟೆ ಕೊಡುತ್ತೆ, nitrogen base ಕೊಡುತ್ತೆ, phosphoric acid ಕೊಡಲ್ಲ. Term enzyme was coined by Curie. All of the following enzymes are protein except trypsin, pepsin, stapsin, except. So trypsin is involved in protein metabolism. Okay. Trypsin is the hormone. The non-protein organic factor during formation of which is attached to apoenzyme is called cofactor. Lock and key hypothesis was put forward by Emil Fischer, coastline in dune fish theory. A specific low molecular weight substance such as the product of another enzyme in the chain reaction which binds with the specific site of enzyme different from its substrate binding it. So what's the question they're explaining here? It's a low molecular weight substance. Okay, it is a product of another enzyme in a chain reaction. It binds to the specific site of the enzyme different from its substrate binding site. They are called as competitive inhibitor. They resembles just like a substrate. Sulfur drugs control bacterial pathogens Okay, by controlling bacterial pathogens by feedback inhibition. Cyanide kills an animal. It inhibiting the cytochrome oxidase, a mitochondrial enzyme essential for cellular respiration. All the examples of feedback inhibition. All are the examples of all are the examples of feedback inhibition except inhibition of succinic dehydrogenase by malonate, inhibition of hexokinase by glucose 6-phosphate, inhibition of phosphofructokinase by ATP, inhibition of threonine deaminase by isoleucine bacteria. Feedback inhibition is inhibition of hexokinase by glucose 6-phosphate. Which of the following group of enzyme catalyzes the cleavage of specific covalent bond and removal of groups without hydrolysis? It's called lyases. It's called lyases. Alcohol dehydrogenase enzyme also exists in isoenzyme. Number of isoenzyme forms of alcohol dehydrogenase. So alcohol dehydrogenase is a type of enzyme that is appearing in different forms. Okay. The number of isoenzyme forms of alcohol dehydrogenase in maize are 4. All enzymes of human body have same pH, optimum temperature, same pH, different optimum temperature, different pH, same temperature, different pH, different optima. All enzymes of human body have different pH, different temperature. A competitive inhibitor of an enzyme increases the Michaelis mental constant without affecting the velocity, decrease the Michaelis-Menten constant without affecting the velocity, increase the Michaelis-Menten constant without affecting the Kmax, decrease both Vmax and Kmax. A competitive inhibitor. Okay, this competitive inhibitor, it is always going to reduce or inhibit the presence of the substrate. So competitive inhibitor of an enzyme always increases the Michaelis-Menten reaction constant without affecting the velocity. A non-competitive inhibitor of an enzyme decreases the Vmax without affecting the Michaelis-Menten constant. An organic substance bound tightly to an enzyme, which is essential for its activity, is called as coenzyme. Which one is most abundant protein in the animal world? Collagen. For its activity, carboxypeptidase require zinc. Most abundant intracellular cation, sodium. Macromolecule chitin is a phosphorus containing polysaccharide, sulfur containing simple polysaccharide, nitrogen containing polysaccharide. N-acetyl glucose amine ಅಂತಂದ್ರೆ it contains the nitrogen. So if you observe this graph, A explains activation energy with enzyme. This B explains activation energy without enzyme. Endothermic reaction with energy A in the presence of enzyme and B in the absence. Is it an endothermic reaction? Okay, so this is a process which is going to explain about the type of the reaction. Activation energy increases the kinetic energy of the system and brings about the completion of substrate into the products. So here, this is a with the release of energy and the ಹೇಳಿದ್ಮೇಲೆ it's a exothermic reaction with energy A in the presence of enzyme and B in the absence of enzyme. Concavalin is a lectin. Purines found both in DNA and RNA are adenine thymine, adenine guanine, guanine cytosine, cytosine and thymine. It's adenine and guanine. So by this, we are going to end up with the session. Okay, we discussed with a few sets of questions regarding to the biomolecule. In the next class, we can continue with the physiology concept. Okay, thank you.